All right, guys, sorry for that. Just making sure that I'm up and going. So today I'm going to be talking about muscle physiology and the physiology of muscle contraction. We're going to talk about energy that's used in that process, about muscle contraction itself, and then the cardiovascular system. These are some references if you'd like to check them out. All right, energy is defined as the capacity to perform work, and work is the application of force through a distance. Now, when we're dealing with body joints, we're not dealing with strictly work. We're dealing with a rotational type of work called torque, and we'll talk about that more later when we talk about biomechanics. You'll love torque though because it involves dusting off your trigonometry skills and using those a little bit. Again, we'll get into that later, but just a preview of what's to come. So torque is a type of work that's rotational. And here are some examples of types of energy. There are quite a few different types. And in terms of safety, I'd add a couple of pneumatic and hydraulic, but in ergonomics, generally, we are most concerned about these two, chemical and mechanical, really mechanical. But we will talk about some forms of chemical energy as well today. All right, so most energy is consumed in biological systems with ATP being broken down from its 3-phosphate form down to 2 phosphate molecules and then a single inorganic phosphate molecule. Most of that transfer is from chemical to mechanical. So you'll see that the bonds between these elements and molecules has stored chemical energy. And when we break this bond, we're actually pulling out the energy and using it, and in this case for muscles, using it to convert it to a mechanical type of energy. ATP has to be regenerated through resynthesis, meaning we break it apart, and then if we want to use it again, we got to turn it back into ATP. And so there's a process for that works best when we have enough oxygen. That is called aerobic work. So when oxygen is available and present, then our body will use oxygen in that regeneration process of ATP. When it is not, then it's called anaerobic work. It does not require oxygen, and it is fast, but there are some downsides to that. Let's talk about those. So it also depends on the type of work or energy that we are talking about. So there could be bursts of energy, so that's quick releases. A lot of times with anaerobic systems, we may also have prolonged work. Usually it's slower, but we're also able to a lot of times get our, catch our breath. And so it's often aerobic work where we're able to breathe enough oxygen and get enough to our muscles that we don't have some of the negatives of the anaerobic systems. And so to talk about those here, let's look down at this table here. I want you to look at these columns here. So you can see for anaerobic systems, we've got 2 of them, this ATP-PC system and this lactic acid system. And for each of these, no oxygen is required. They're both relatively fast. The problem is they're not that efficient. You can see that there's relatively little, let's see if I can hide that there, relatively little ATP produced. However, with an aerobic system, and you can see here, this is what we usually think about when we're talking about calories, right? We've got our carbohydrates, our fats, and our proteins. O2 is required. It takes more time, but it's much more efficient. And we're not going to break down these. This is not a nutrition class, but carbohydrates are the most efficient of any of these, and that's why, at least according to the current science, current train of thought, you're supposed to avoid carbs if you want to train your body to eat itself, basically. So over time, we can develop an oxygen deficit. If you look at the graph down here, you can see this time scale. If we were to start exercise or workout, you can see the oxygen deficit building over time for the amount of oxygen we're lacking, but we need. And we get to a point here where it's a complete deficit, excuse me. We stop our workout, our burst of speed. This is quite the workout, though. It's more than just a couple minutes. We've almost got several minutes here. We finish our set, maybe all of our reps in our set, and we stop. And so the body's able to slowly deplete that deficit, or rather decrease and reduce that deficit to where we have no more oxygen debt. Again, we have systems that are designed to deal with a lack of oxygen, but some of the main drawbacks, the accumulation of lactic acid. So if we go back here, we can see this lactic acid system is, it's a common way for our body to deal with anaerobic situations, and it produces lactic acid, which is painful. It doesn't feel good. That's why they say, no pain, no gain. That's why that saying exists. It's because of the lactic acid building up in our muscles, and it doesn't feel good. All right. With muscle contraction, we've got areas of the brain, or the motor cortex, that are designed to control different parts of our body. And it's interesting and amazing, I think, that we were able to map these different areas from the cortex to our body. But you can imagine this signal will leave the cortex and travel, that's not what I meant, travel down the spinal cord through efferent nerves. So any signal going away from the cortex towards muscles will use efferent nerves rather than afferent. We'll see a little slide on that in just a minute. And then it ends up in our skeletal muscle. A homunculus is a representation of that mapping in the cortex, where parts of the body are larger, different features are larger for those that have the largest map on the cortex, and others have the smallest. So you can see here, there's not a lot of brain cells, of neurons, dedicated to moving the wrist, or at least the end. That may not be the wrist, that may be representing the end of the forearm there, but there's not very much. You can see, for the legs and the feet, there's just not a lot there. But we have entire sections of our brain dedicated even to fingers here. You can see thumb, index finger, middle, ring, and little, the pinky, and the hand as well. All have quite a bit dedicated to them, just to them. And that's why you see such a large hands. We do a lot with our hands. They're very important. The sort of precision and finesse that humans are able to achieve with hands is pretty amazing, actually. You can see there's also a lot for the lips and face. Speech is important to us, and facial expressions, and so that might be a reason why that is the case. We also have a homunculus for sensation, and you can see there are not a lot of differences. Again, we still have the hands dominating this figure. Some in the lips again, maybe the second largest there once more. And you can see some of the other areas have grown a little bit in size. The forearm doesn't look quite as tiny as the last figure. But just to get a sense of what parts of the brain may be sensing things from different parts of the body. All right. For nerve conduction, we have to have an initial stimulation to get this process going. Now, when talking about muscle contraction, that would start in the brain, in the cortex, the area we were just looking at. Depending on what part of the body we're moving, it would be a different part of the brain. And we're going to talk about how that happens exactly. So separation of positively and negatively charged ions in our body creates potential energy. And in the cells, we make this separation with membranes. So you can see here, this is the membrane within a neuron, this light blue thing here. And you can see there is a difference between the charges inside, the yellow is inside, and outside. So the difference in the potential energy between across this membrane is called membrane potential. And like electricity, we actually measure it in volts. But it's a much smaller amount of electricity than what you might find in your house. So it's a much smaller number. But you can see here when your neurons are resting, that membrane potential is at negative 70 millivolts. So you can see it's not a zero. We do have a difference here. It's kind of like batteries. They each have a positive and a negative side, and because of that, the opposites attract, and the positive ions are trying to. Actually, what happens is the negatively charged ions are going to move electrons through whatever circuit, whatever system you have, to the positive side. So that's how batteries work, and that's why eventually their charge is depleted, because it's an electrochemical gradient. And that is actually, funny enough, that is what we have here. We have an electrochemical gradient where within the cell we have potassium ions, so that's these yellow ones, and outside of the cell, outside the membrane, we have sodium ions. Now you can see they're both positively charged. So how is it that we create a gradient here? Well, there are fewer potassium cells inside than there are sodium, ions outside. Also, these negative charges you see here are proteins within the cell that happen to be negatively charged. So the combination of this environment creates a charge of negative 70 millivolts inside compared to the outside. And that is called the resting potential for this neuron. So the neuron membrane is lined with sodium potassium pumps, and we'll talk about that a little bit more in a minute, but those pumps are designed to move these around. So when we have let's come down here to this lower figure, step 4. So when we have this this imbalance where the membrane, the neuron is no longer resting and it's already been depolarized, we need to return conditions to the way they were. And so a sodium potassium pump will move for every 2 potassium ions it moves in. It also moves 3 sodium ions out. So this little pump is going to work on creating that difference to maintain that resting potential, the membrane potential. So that pump is not always working on that, but if there is no stimulus from any other part of the neuron or any other source, then it's going to be doing this, trying to create an imbalance. If you are familiar with the body or even just nature, you know that generally it's trying to get back to a state of balance, an equilibrium, where it has the same number. If this membrane wasn't here, there'd be the sodium and potassium ions would intermix, and they would be fairly evenly distributed. It's amazing how nature finds that balance. It tends to always come back to that. This membrane and these channels and the sodium ion pump, sodium potassium pump, are designed to create an imbalance. It is natural, but it's kind of artificially, I guess. So they create an imbalance to be able to create that potential energy that the neuron is going to use. So, and it's also, I mean, you could think of it not just like batteries, but like magnets where the two opposites are attracted. It's always, the positive is always going to be trying to reach the negative. And if in this case, we have to create that separation with the membrane. All right, so real briefly, the membrane has these channels. So this is a sodium channel. Here is a potassium channel. Those channels are triggered to open when the membrane potential, resting or membrane potential of the neuron's membrane reaches a certain state. So here it's showing it at negative 60 millivolts. Some people say negative 55. It's somewhere around there, around negative 55, negative 60. If there's a stimulus and you have this charge moving down, I'm on the step 1 figure here now, moving down, you see that red. You have a charge incoming from further down the neuron that is, it's called local current here, just like with electricity. Yeah, I mean, this really is an electrical signal. But anyway, so that current kind of moves in. You can see further down the line that something was going on, and these sodium ions are kind of moving in, and they're changing the potential here. It's actually getting it closer to zero. When that happens, that triggers the sodium channel to open up, and the sodium ions move in. As that happens, we also get later, we are going to get activation of the potassium channels, and they'll open up to further depolarize the situation. Not yet though. So right now we have a bunch of sodium channels, and they all open up, and you get this huge influx of sodium ions. So now you can see that we've been depolarized to positive 10 millivolts. So many positively charged ions, these sodium ions, have come in that now it's positively charged. So there's more positive charge inside than there is outside. Once we reach this depolarization of plus 30, so many have come in that now we're at plus 30. It triggers the potassium channels to open up, and the potassium ions leave. Some people say this is more at positive 40, but it's somewhere around there. Basically, though, once we reach this stage, then we have reached what's called an action potential. So before we reach that stage, no signal will be sent. I'm not going to spell out potential there, but we have to reach this for there to be an action. If we can reach this of plus 30 or plus 40 millivolts, then the signal will send. It will continue down the line. If not, then this impulse will kind of die out, and it'll never reach the end. So if you think about something that maybe brushes up against your skin and triggers a stimulus, but it's not big enough to create the action potential, it doesn't reach plus 40 millivolts, the signal is never going to go to your brain, and you're never going to know that that thing brushed up against your skin. So it's got to reach that action potential, and then we have action. We got it moving down. Anyway, so this area of the membrane, we sent our signal. We reached plus 40. So now we got to come back. We got to repolarize the membrane. So what happens is that sodium potassium pump, like I talked about earlier, will start moving the ions back to where they're supposed to be, so that we can get back to this stage. And in this case, actually what happens is the pump starts working, and it over polarizes the membrane a little bit. And I'm not sure the purpose of that, maybe just to be sure that it doesn't get overstimulated. But anyway, so that's what happens. Then eventually it returns to the resting state of minus 70 millivolts. We talked about a motor unit, and remember, muscle signals will come from the brain and go out to the muscles in the efferent nerves. So you can see it's the ventral root. So I want you to think about what side of the vertebrae is that. So this is either facing forward or backward, and this is facing forward or backward. Think about it for just a minute. Ventral versus dorsal. So if you remember our little review of anatomy, ventral means on the front and dorsal means on the back. So this would be towards your guts. So these nerves come out on the in the abdominal or thoracic cavity on that side as they travel down to the muscles. Sensory signals and impulses come through the afferent nerves and travel up to the brain. So any sensory information is going to come in. Sorry, I drew that wrong. It's going to come in this way and go up to the brain. So you can imagine that might have interesting ramifications for, if you have damage on one side of the vertebrae but not the other. Maybe you can't feel, but you can still move, certain parts of your body. Anyway, so that's how that's organized. And a single motor unit. So we have these neurons coming in from the spinal cord into a muscle fiber. A single somatic neuron. So a somatic neuron is one that goes to muscles, can innervate, meaning it can be attached to and stimulate multiple fibers. And usually it only gets down to like 3 as the minimum. And it could innervate up to 1,000 muscle fibers. So I want you to think for a minute, what muscles in your body might have only 3 fibers innervated by a single neuron versus 1,000. So if you fire that neuron, in the case of the 3, only 3 muscle fibers are going to move. They're going to contract. Whereas the other one, if you fire that neuron, 1,000 fibers may be going off. So if you guess that the 3 may be in the hands or somewhere like that, then you're right. So for fine movements where you just want to move just a little bit, that's where you're going to have motor units that only include 3 or 4 or 5 muscle fibers, whereas motor units, say to your glutes or your quads, they're probably going to innervate hundreds, maybe even up to 1,000 muscle fibers at once. So that when that neuron fires, that motor unit fires, then you've got a bunch of stuff going on all at once. So quick review, we've got the muscle, then the muscle fascicle. So that's this thing right here. It looks kind of like a, I don't know, a sausage. Anyway, we got a fascicle, and within a fascicle we have a fiber, a little sausage. And within the fiber we've got a myofibril, and within the myofibril we have myofilaments. And then sarcomere is going to be an important thing. So that's one functional unit within a muscle. And we've reviewed the bands. Just know what those are. All right, some quick anatomy, microanatomy for myofilaments, and then we'll talk about the physiology. So the thin filament is this blue filament here, the actin. And the thick filament is this one right here. Looks kind of like a muscle. It's same color as steak. This is called myosin, and these heads here are called the myosin heads or head groups. You can see it's got two heads in each of these. Another important part are these 2 proteins here, tropomyosin and troponin. They are an important part of what we're going to be talking about in terms of contraction. This is a quick overview of that process. So first we have the neuron's impulse that comes down across the synaptic cleft. We'll talk about that. that causes secretion of this neurotransmitter acetylcholine. Excuse me. So that crosses the gap and stimulates the muscle fiber, creating an electrical signal within the fiber itself. So it's in a way, it's kind of like a neuron, because it can transmit that signal through itself. So again, remember, one motor unit is the nerve plus all the fibers that nerve innervates. So as the signal is going down into the muscle, we don't want the acetylcholine hanging around, and so acetylcholinesterase gets secreted, and it does kung fu on all the acetylcholine. It breaks it down, and then it gets recycled and reused. But that process deactivates the acetylcholine and makes sure that you don't overstimulate the muscle. All right, to look at what's going on at this cleft here. So you can see here kind of the depiction of the nerve impulse traveling down the membrane. Now it's supposed to be negative. Remember, a negative charge inside compared to a positive charge outside. But you can see, at least in this portion of the that portion of the synapse, it's reversed because the electric signal is here and it's activated. So once that signal is sort of a wave, moves down here, kind of like a ripple moving through the neuron's membrane, once it reaches here, it opens up some some calcium channels, and calcium comes in to the cell. That influx of that positive charge causes these vesicles that hold acetylcholine to move out. They don't want to be in there, apparently. Maybe calcium is a lower class citizen or something, and they don't want to be in there. Anyway, so the acetylcholine moves out into the this synaptic gap, and then it moves across and binds with these receptors on the muscle side, and that binding action creates another impulse. It moves kind of like a wave, and that moves down the sarcolemma. So it's basically just the membrane of the muscle fiber. So we'll talk about where that goes next. But real quick, there are a lot of drugs act on neurotransmitters or on the synapse in that area of our bodies. You know, whether it's prescription drugs or illicit drugs, they they'll act there. So, for example, let's go back here real quick. If we were talking about not muscles, but in our brain, neuron to neuron connection here. These sometimes in some nerves or synapses, acetylcholine would move back. Once it's done doing its job, it would move back and fill in a channel to get ready to be used again. And it wouldn't be acetylcholine, it would be a dopamine. So this is with cocaine. So what cocaine does basically is it comes in and it will plug up this channel so that the dopamine can't go back into the synapse. So you get a bunch of dopamine hanging out, in this gap, continually stimulating whatever the nerve is on this end, making you feel good, but then also all the negative side effects that come with that. You can get kind of jittery or paranoid. Anyway, so basically, dopamine starts building up like crazy in here, so you feel good, but then there are the negative consequences. So some drugs act on this area. So these two will block acetylcholine from doing its job. So botulinum, it's in honey. It's why infants aren't supposed to, less than 1 year of age, not supposed to eat honey. Adults, we can handle it. Our digestive systems will take care of it. before it's even an issue. Then curare, it actually blocks, so it blocks the receptors on the muscle fiber membrane, meaning if you did that, you'd either of these, you would not be able to contract your muscles. This one blocks the release of acetylcholine. And then you could block the enzyme that breaks down acetylcholine, and that would be malathion and parathion. They are pesticides. If acetylcholine blockers prevent muscle contraction, then what would an acetylcholinesterase inhibitor do? I want you to think about that for just a second here. So if these, because they block acetylcholine, they block contraction, what would this do? So you can imagine these would cause prolonged contraction because you can't break down, kind of like what cocaine does with dopamine. So we can't, the acetylcholine will just build up in that gap and continually stimulate the muscle fiber to continue to contract. Not good. You would not want that. All right. So that impulse moves down the sarcolemma. It's just the membrane for the muscle fiber, fancy name for that. Moves down through these tubules, and you can see it starts out negative, but then once we've got stimulation, we get positive. This change here causes calcium to some calcium channels to open up, or vesicles that open up, and they dump calcium into the sarcomere area. So let's talk about that. All right, so I mentioned troponin, tropomyosin. So there are proteins that bind onto actin, and actin, they block these binding sites on actin. Here we have actin. We've got the myosin head. So ATP comes in and it binds with the myosin head group, and that binding kind of loads it like a spring, so it pulls back and it's ready to go. It really wants to bind with actin, but with troponin and tropomyosin here, it can't. It's blocking the sites, so it's got to wait its turn. So that requires ATP to load it, basically. And like we talked about, the energy really comes in the chemical bonds within this molecule. So what happens then, we talked about calcium coming in. So these little dots are the calcium ions. So calcium comes in, and troponin and tropomyosin really like calcium. So they release from the actin. They move out of the way, basically, of the binding sites, because the calcium is keeping them occupied. And once that happens, then the loaded myosin head springs forward and grabs onto the actin, the thin filament. And that action, you can see it down here, that action pulls the actin filament across. So you can see the Z band was here. Now it's moved up a little bit. So it is kind of like pulling a rope with your hands. Really, that's what it's like. So that process used up the energy in the chemical bond of this one little phosphate to the ATP. And so now it becomes ADP, diphosphate. And then it gets released. So after that happens, the ATP pops off, and the myosin head is, it's attached to the actin, hanging out. It's where it wants to be. To get it to release, you have to have another ATP molecule come on in and attach to it. So that's where we talked about rigor mortis a little bit, as an example. So if you're dead and there is no more energy in your body, there are no ATP molecules that come in and make that release, and that's an issue. If you're dead, maybe it's not that much of an issue, but there are other diseases and disorders where that is an issue. Anyway, so you have to have another ATP molecule come in to actually make the myosin head release, and then spring load itself back, like the top picture. So it gets spring loaded again. And if there are binding sites available, then it will release and bind with actin and pull it again. If though that nerve stimulus has stopped, the impulse and the signal has stopped saying, hey, contract, then there are calcium pumps. And I'm just going to draw one real quick. Little calcium pumps that are grabbing all the calcium and taking it back out of there, taking it out of the sarcomere. So if the nerve impulse has stopped, those pumps go. And once they've got enough calcium out of there, then you're going to get back to this resting state, like at the top figure, where there's no calcium. That troponin and tropomyosin bind back to their sites, and they block the myosin from doing what it wants. So hopefully while you're resting, but maybe if you're in anaerobic conditions and I don't know, you're sprinting or doing a lot of powerlifting and I guess, I don't know, there are a lot of things you could do that would be anaerobic. But anyway, you don't have enough oxygen. Either way, though, your body's gonna be recycling that ADP and the phosphate ion, the inorganic phosphate, and making it back into ATP again. So that takes energy. That takes calories. So that's why if you're doing any sort of work, running, working out, using your body, your muscles, that uses up calories, uses up energy, and that's where you can burn some fat off if that's what you want. But anyway, so let's see, and I think that's the rest of that one. There are a couple of different types of work: dynamic and static. So dynamic is where we're moving around, and to be honest, that is what we are designed for. So we contract, we relax, we contract, we relax. We're doing different things with different muscles, and that keeps blood flowing. It keeps things flowing. Blood has nutrients, and it carries away the trash. That's really important. For static work, we have prolonged contraction of muscles, and that is bad, depending on the length and the muscle. But if you have prolonged contraction, then you get buildup of waste in your cells, and they can't get the nutrients and enough oxygen, and so you start, it hurts. You get some lactic acid buildup maybe, and it just doesn't feel good, and that's because of what's going on. So when you're resting, think of this as a little table here. When you're resting, you don't need a lot of blood, and there's not a lot of blood flow going on because you're resting. We'll talk a little bit more about where your blood goes, but in a dynamic work, you need a lot of blood, and you're getting it, and that's the key there. The key difference is these two right here. With static work, you still need a lot of blood, but you're not getting it, and that is the problem. You get waste build up, and your muscles, your body is not getting the nutrients it needs. Just some examples of static work. An assembly line, you don't want that job, but you may work for a company that has that, and if you are in charge of ergonomics and safety, that is going to be an issue that you're going to be working on. Sitting at a computer, very static. Some hand tools are not designed well, where you have to continue to apply force. Hopefully you have tools where there's a little lock or something, you can lock it in in position. Standing in one place, not good. Most people like to shuffle around a little bit, shift their weight on different legs. That's all to try and be dynamic and get the blood moving. Also, the more that you have, more force you have to apply, the more the blood flow is going to be constricted. You'll also use up more energy, increase your heart rate, and you'll need a longer rest period just to recover from that. So if you have a muscle that is not getting enough blood flow, it could get sore, kind of cramp up. That is called ischemia. And usually it's short term. Hopefully it's short term. But like I mentioned, waste products start to build up, like lactic acid and potassium, and you'll get pain. You'll get pain. So you can see here this graph. The y-axis is the contractual intensity, and on the left, the y-axis on the right is the pain intensity, and then here we have time in minutes. So you don't, according to this graph, with occluded circulation, you'll start to have pain at about 25 seconds. I'm guessing right here would be 30. So we've got 25 seconds, you start to feel pain. It starts to get real high and kind of peak out here after about 2.5 minutes, 2 minutes or 2.5 minutes. That's the contraction is still going, but then you let go, and it's going to take a while for that pain to subside. So you can see you have about 40-45 seconds where you're not contracting anymore, but you still have pain. You can see here where when circulation is intact, that sustained contraction, it's not quite as painful. Peak here, maybe 6, but it's only for a moment, whereas you have a sustained 7 when you have blood flow occluded, when you have ischemia, not enough blood getting there. So again, static work is bad, dynamic work is good. All right, the cardiovascular system. Think of the heart as a pump. That's what it is. It's a pump. Think of your arteries and veins as kind of like the halls in your house or in a big building. Their only purpose is to transport stuff. Like nothing functional actually happens except transportation in there. And then think of the capillaries as, I don't know if we're doing your house or building, then maybe it's the office. So those are the key parts: the heart, the transportation system, which is your arteries and veins, and then the capillaries. So the heart is really interesting. It's divided into 4 compartments, and the compartments on top are called the atria. So we have an atrium on the left side, an atrium on the right side. I think of attic. So it sits above the ventricles, so it's on top, and it receives incoming blood. It's sort of like the waiting room. Let's start with the left ventricle here, and we'll go through the whole cycle. So the left ventricle is the biggest, strongest part of your heart because it's got to pump blood from the heart to the rest of your body, basically. Not everywhere. We'll talk about that in just a minute, but pretty much, from your head to your toe, it is pumping blood to all those places. So it's got to be strong, and it's big. So it pushes blood, and so you have your arterial blood, so arteries, arterial blood moving towards, now in this case, we're talking about muscles. So it's going towards the muscles. Once it's divided into all its little branches and it gets down to the tiny, tiny capillaries within your muscles, then it's going to be dumping the oxygen that it has. And I say dumping, I mean it's kind of like what we've been talking about. There's this membrane, and there's more oxygen in the blood than there is in the muscle, and so the oxygen just wants to leave. So the oxygen takes off, but the opposite is happening with CO2. So inside the cell, there's a lot of CO2, but in the blood, there's not. This is nice, clean blood coming from the lungs. So the CO2 in the cell, in the muscle, it wants to bail. It doesn't like it in the muscle anymore because there's a lot of CO2. It wants to kind of balance out. So it jumps and bails into the blood. So then you get venous blood. So your veins will take it back to the right atrium. So this is the left ventricle, left atrium. We've got the right atrium, the right ventricle. So the right atrium may be one of the smallest, weakest, maybe the smallest, weakest part of your heart. I'm not totally sure which is weakest. Just know that left ventricle as the biggest. So it's kind of the waiting room. So the blood fills, I mean, it hangs out. It's not for very long. It's just milliseconds. I mean, it's just not even a second that it's going to be hanging out in there. Then this valve will open and it will come into the ventricle, and that will fill up. And the right ventricle will then push that blood to the lungs. Now, this can be kind of confusing if you ever hear something referred to as, well, so any vessels that carry blood away from the heart are considered arteries, and vessels that carry blood back to the heart are considered veins. If you remember that, then you won't get confused. So this is a pulmonary artery, but it has blood that's blue and deoxygenated, like you think of in a vein. Okay, but just remember, anything that's going away from the heart is a vein. Or excuse me. Oh, I hope I didn't make that worse. Away from the heart is artery. Away, artery, and back to the heart is vein. So the pulmonary arteries carry this deoxygenated blood that came back from the rest of your body, and it's got a whole bunch of CO2. So the CO2 bailed, and the oxygen also bailed, but back in, into the muscles. So we got a bunch of CO2 and a little bit of O2 coming into pulmonary capillaries. And again, that's the functional part. That's where the action takes place. Our lungs hopefully have a bunch of oxygen and not much CO2. And so the CO2 sees that and thinks, hey, I want to be where all that oxygen is. So the CO2 bails and it's going to get breathed out. The oxygen in the lungs sees that there's not a whole lot of oxygen in the blood, and it says, hey, that's where I want to be. So it comes in. This is in the alveoli. That's where the capillaries come in. The oxygen says, hey, I want to be in that blood. So it jumps in. So now we have nice oxygenated blood with not much CO2. I mean, very little. Coming back in, it enters the left atrium. Again, the waiting room starts to fill up, and it's waiting for the blood that's still in the ventricle to get pumped out. Once that gets pumped out, then it comes into the left ventricle and gets ready to be pumped out again. All right. So the alveoli, again, is where the capillaries come in. That's where the action takes place. This anatomical dead space, it's like the hallways. It's not used for anything except the transportation part. So for your lungs and your airways, it's the same. So your nose, mouth, pharynx, larynx, trachea, all these areas, nothing happens. It just moves the air. That's all it's for, just like the hallway. The only air that's going to actually, see any action is the alveoli. Yeah, anything that gets down into the alveoli. So a few things that affect alveolar ventilation is the depth of breathing, the rate of breathing, and the size of the dead space. So hopefully you don't have a lot of dead space, and it's pretty efficient. This is just showing kind of what we talked about already, but in a more simple diagram. So when we breathe air in, the inspired air, we got a lot of oxygen. This is partial pressures. A lot of oxygen, not very much CO2. So that comes into the lungs. This is supposed to be the trachea. This is in one alveolus. And you can see the blood coming in, this venous blood, it's almost got 50% oxygen. It's got a little bit more CO2, but compared to what's in the lung, proportionally, it's got a lot of CO2. So the CO2 bails into the lungs, into the alveoli, and the oxygen in here is like, hey, that's where I want to be. So it comes into the bloodstream and moves through. Now you can see we went from about 50-50 to now, almost 3 times as much oxygen as there is CO2. So that then travels to the muscle where that exchange happens, and then we get back to that 50-50, and then we come back around. And that's a more simplified version. So we talked about this. Oxygen, so for hemoglobin, it's divided into 2 parts. You have the heme part and the globin part. Oxygen likes the heme part, and CO2 likes the globin part. So that's nice. It's kind of like a shuttle that carries two different kinds of passengers. It can handle it all. So we talked about this a little bit. 2 muscular pumps. Those pumps are the ventricles. The atria are they have some muscle, but they're not really for pumping. They're more of the waiting room. Again, the left ventricle is what pumps to everything pretty much. The right ventricle pumps to the lungs. So you can imagine, it's like its next door neighbor. It's not very far. It doesn't have to work that hard to just pump it, across the street. Whereas the left pump, it's got to, again, pump it to everything from the top to the bottom. So it's big and it's strong. All right, so they contract together as one fiber, which is really interesting. It's an all or nothing law, just like a nerve impulse. So it's all going to go or none of it's going to go. It is similar to skeletal muscle or striated muscle, but it has its differences, and that's why it has its own category. And it's autorhythmic, meaning the rhythm is produced right within the heart. We'll talk about that. So I want you to look at the picture here on the right, and we see our heart. So we see you can kind of identify some of the compartments here, the chambers. So we got a right ventricle here. Right atrium, right ventricle. Left atrium is kind of it's kind of back behind here, and then our left ventricle, the big strong pump for our heart, is right here. So you can see a lot of this electrical circuitry that we're looking at, the kind of purple lines. It's mostly on the right side, but it goes everywhere. It's got to innervate all the entire heart to get it to do its job. So we've got a couple of nodes. So this is the sinoatrial. It's on the atrium, so that makes sense. Sinoatrial node. And then we've got the atrioventricular node. So you can see if we were to split this right here between the right atrium and the right ventricle, this one is kind of on that line. So that's why it's called the atrioventricular node. Sorry. All right. So what happens is the rhythm, the electrical impulse begins up at this SA node, the sinoatrial node, and that spreads down through these pathways. This is getting difficult, so maybe I won't annotate. But anyway, you can imagine it travels down these pathways to the AV node, the atrioventricular node, and then that sends it down to the rest of the muscle and activates it. Again, all kind of simultaneously. It has to work together. It's important that it works together. So a lot of cardiovascular diseases, heart diseases, and disorders involve, if you know what a pacemaker is, that's what it's designed to do, is to synchronize that better, because for some reason it's off. And so that signal travels down to the ventricles. That's the important part. The pumps are the most important parts, the ventricle on either side, that activates them, and they pump simultaneously. Or each side does. So you saw here recorded by EKG. So EKG is the old school term. Now, everyone refers to it as an ECG, electrocardiogram, or cardiograph, either way. So this is an example of the output of an ECG. And you maybe have seen, some of those shows like, I forget what they're called. I don't Grey's Anatomy, maybe. I don't know, those shows that feature healthcare settings and healthcare professionals. I forget what, there's some famous ones. I never watched them, obviously. But anyway, or most movies, you may have seen an ECG screen where when this line becomes flat, then everyone knows that the person's dead. So that the reason that we know they're dead is that it is measuring electrical impulses in your heart, so that across the SA node, the AV node, and down the pathways, that is what it's measuring. So there are different waves. So we've got the P wave. That's that first little wave that you see. That represents depolarization of the atria. So we can go back here. Starting up at the top. Hopefully I can do this. It's getting kind of crowded in here. But anyway. So starts up at the top and moves down the pathways. All right, sorry about that. Moves down the atria, down these pathways. All right, next, depolarization of the ventricles. So the QRS, that's this whole thing here. This little drop, then a spike up to R, then back down to S. So that is depolar. That's the big thing, right? That's what you're wanting to happen, is for those pumps to go. Oops. So that represents it reaching down here and depolarizing ventricles, meaning it's going to stimulate them to pump. This just gives you an example of the time this takes. So it's just over, it's under 0.2 of a second after the P wave. So now I don't remember for sure, but I would imagine that depolarization is primarily represented by the R peak here. This is like we saw with the neurons. Sometimes it kind of over adjusts, or like with hyperpolarization, when it repolarizes, it goes a little bit farther than it really needs and then comes back. So I think that's what's happening here, is with the Q and the S, is you're getting repolarization of the atria. Well, that's not detectable, but you may, on this S, as it comes back, as those pathways in the ventricle are repolarizing, getting ready to go again, it may overshoot. Just like in a neuron, it gets down to negative 90 millivolts, even though at resting it's supposed to be at negative 70. So I believe that's what's going on here. And then the T wave is the repolarization of those ventricles. All right, so that's what you see on an ECG. Now, to finish up here, we are going to talk about that the distribution of blood in our bodies. I want you to think of a time when maybe you ate right before you went running or going to work out or swimming. Let's say you had a big meal and then you were trying to do something aggressive, like you were really working out, or you're going to go on a long run. You probably got cramps. and so the main idea here is we've got this figure here, but it is when you eat, the food passes into your gut, and your gut is saying, hey, I need blood. I need I need you to come get these nutrients and clean out some waste. And and that's part of the reason why you get sleepy after a big lunch. Let's say you eat a big lunch and your gut is saying, hey, I need the blood. Sorry, brain. Sorry, muscles. we got to have it over here. So blood goes, it gets moved, more of it gets moved away from your brain and to your gut. You get sleepy. Now, if you are unfortunate and you tried to swim or work out right after that, then your skeletal muscles are also saying, hey, we need blood. So it's kind of this tug of war between your gut and your muscles, each of them saying, hey, no, we need more. And when they both need more and they're both not getting what they need, then one is going to lose, and whoever loses is going to cramp and it's going to hurt. So this is kind of just showing how big the need for skeletal muscle, for blood, gets in skeletal muscles. So you can see muscle starts out this little sliver right here. But as you start to exercise, it grows pretty fast. You can see, that it gets pretty hungry. It needs oxygen and it needs energy, and it needs you to, your blood to clear out that lactic acid and all those waste products. And so, again, I don't know what this graph looks like for the gut, but you got this competing with your gut. If you had just eaten a meal. And, either way, you're going to be hurting if you've eaten a lot and you're doing maximal exercise. Something's going to lose and it's going to hurt. And that's it for our review of physiology. Again, so just to recap here, we talked about energy and briefly about its use and how it resynthesizes. And if you have oxygen, then it goes slower, but it's much more efficient and it doesn't create as many waste products. If you're in anaerobic conditions, then you're going to have waste products and it's going to hurt. It is fast though, but it's not efficient. With muscle contraction, we're not going to go there, but just remember the process for that neural impulse down the neuron, across the synaptic cleft, into across the sarcolemma, it's the membrane of the muscle fiber, down into the sarcomere. Calcium gets released, the actin and myosin link up, and the myosin basically is like your hand pulling the actin rope. And then finally the cardiovascular system. So you basically get that same sort of exchange of stuff, but it just happens to be oxygen and CO2. So in the pulmonary capillaries, you have CO2 leaving the blood and oxygen coming into the blood. And then as it gets circulated around to the muscles, the oxygen leaves into the muscles, and the CO2, which has been building up because you've been working out, leaves the muscle and goes into the blood so it can get transferred out. Thanks.